Okay, so now we're going to start our discussion on genetics. So I want you to take a minute and think, what do you remember about DNA? Okay, if I put a piece of paper in front of you and ask you to just write down everything you could recall, what would you write about DNA? Okay, and when you're ready, we'll move on. All right, so DNA stands for deoxyribonucleic acid, DNA. We talked about the macromolecules, so there are four. You should remember them: protein, carbohydrates, lipids, and nucleic acids. So DNA falls under nucleic acid. It's literally in the name, deoxyribonucleic acid. What's the monomer of a nucleic acid? Right, a nucleotide. And then what does DNA do? So we know it's found in the nucleus of all eukaryotic cells, right? And it serves as the genetic information of an organism. It makes you who you are. It makes you look the way you look, okay? It codes for the traits and the proteins that you need. Then this DNA is passed down from parents to offspring, okay? This is a nucleotide, so it's a phosphate group, a 5 carbon sugar, and a nitrogenous base. In this case, that 5 carbon sugar is called deoxyribose. And then those nitrogenous bases can be adenine, thymine, guanine, or cytosine. Okay? Adenine, thymine, guanine, or cytosine. Here they are: adenine, guanine, cytosine, and thymine. And we're going to talk about uracil a little later. So DNA is this double helix, this twisted staircase that we're used to seeing, okay? So on the outside is that sugar phosphate. So this sugar phosphate is what you find that makes the outside. And then those nitrogenous bases bound together give you the inside. So here you have it, those nitrogenous bases. These are hydrogen bonded together. And then on the outside is a phosphodiester bond, and that holds together the sugar phosphate backbone. Okay. So it's important to note here that the way that these bases join together is not accidental. There is a method to this. So A binds to T and C binds to G, always and forever in DNA. A to T, T to A, C to G, G to C. Thymine and adenine, there are 2 hydrogen bonds. Guanine and cytosine, they have 3 double bonds. So on the outside, you have your sugar phosphate backbone bound together by a phosphodiester bond. On the inside, you have your nitrogenous bases held together by hydrogen bonds. 2 between adenine and thymine, 3 between cytosine and guanine. Okay? And now the way in which it turns and they're bound gives you major and minor grooves, and that's just the spacing. So is it a large space or a small space? And those grooves are important because they dictate which proteins can interact with our backbone, and that's or with our bases, and that's important when we talk about replication transcription. Okay. Now, we talked about how these things bound together, right? And so remember that C binds to G and A binds to T. So if this is one strand of DNA, you can tell me what the other strand is based on this, how they bind together. So anywhere you see an A, it's going to bind to a T. Anywhere you see a T, it has to bind to an A. Okay, so G binds to C and C binds to G. And then you can finish the rest of this, right? So C always binds to G, G to C, A to T, and T to A in DNA. Okay, so take a second and fill that in. And then once you're finished, come back. And then I want you to again sit and think, what do you remember about RNA? Okay, what could you recall? If this was your quiz today, hey, write down 5 things about RNA. Could you do it? Okay, so take a second, and then when you're ready, start the video again. So RNA is ribonucleic acid. It doesn't have that deoxy, okay? And again, it's a nucleic acid. It's in the name. Still have a monomer. Now these functions are different because this helps DNA to be made. Okay, so RNA helps us transcribe our DNA. It helps in the translation of DNA. It helps in the replication of DNA. Okay, so there's a lot of functions for RNA. It does some targeting and other things as well. So it's also the same nucleic acids here. You have your phosphate group, your sugar. In this case, it's still a 5-carbon sugar, but it's ribose instead of deoxyribose. And then you have 3 of the same nitrogenous bases. So you have adenine, guanine, and cytosine. But instead of thymine, you have uracil here. So adenine, cytosine, guanine, and uracil. And then here's an example of it working. So these tRNA, transfer RNA molecules, are made of RNA. And then mRNA, and then the ribosome. So here we have tRNA, the ribosome, and the mRNA, all functions of RNA. The structure is again similar, but it's a single structure now. Okay, it's a single line. So here's our RNA. It still has the sugar phosphate backbone held together by a phosphodiester bond. Okay, now I said it's a single structure, right? So what is it hydrogen bonding to? So remember that the RNA can do 2 things. One, it forms this hybrid molecule with DNA during transcription. And then 2, it can fold onto itself. So when it does this now, A binds to U, T will bind to A, and then C and G still bind together. Okay. So if I give you this, you have to remember that if you see an A, you put a U. If you see a T, you put an A. And then C and G is the same that you've seen before. So take a second and fill this out as well. And when you're done, come back to the next video.